Hello, yeah. Hope that uh, my voice is audible. Yeah, let's talk about all those things. Ah, okay, fine. At the night, we'll talk all those things in related to the enzyme. So this is a broad topic. Hope that you guys can hear me properly. So it's a kind of small class, and so in that class we will just cover some of these key important concepts and basically the enzyme kinetics. So few important aspects like okay, I'll just take, yeah, now camera is on. Yeah. So basically, first some of the metal ions and they are cofactor for the enzymes. So like Cu2 plus, it's inside the drops. Yeah. So I think some hang issue is there. Yeah. So okay, let's check my pen once. Yeah. Why it's not responding? Yeah. Now it's responding. Okay. Now it's responding. Uh, Delete it. Yeah. So you know about the copper ion. What happened with it? So So hope that you guys are seeing me. So today I'll kind of explain about the enzyme kinetics and some of the important aspects and some of the cofactors and their activity of different types of enzymes and what are the famous kind of things. And so let's start it. So firstly, I'll just mention some of these like copper 2 plus. So these are the cofactor in the enzyme of the cytochrome oxidase. So you can write down. So the enzyme cytochrome oxidase, the copper for the cytochrome oxidase, and then their important is K plus. K plus ion is the cofactor of pyruvate kinase. Pyruvate kinase. So these are the enzyme. Uh, these are the enzyme and their cofactors. So in that line, you can say cofactor, and these are the line like Cu to cytochrome oxidase, Cu plus copper ions. This is the cofactor of the cytochrome oxidase. Now let's talk about the Mg2 plus magnesium ion. So it's a cofactor of hexokinase and glucose 6 phosphatase. Glucose 6 phosphatase. So like copper plus copper, calcium, magnesium, and along with that the manganese Mn2 plus, that is the important enzyme of the arginase, and ribonucleotide reductase, and nickel plus Ni2 plus, this is the cofactor of the urease. And some of the another important ion is the Zn2 plus. This is a zinc ion, and this the cofactor of the carboxypeptidase A. This is the carboxypeptidase A and and B. So these are the cofactors that are these are the metal ions, acts as a cofactor of that specific enzyme like cytochrome oxidase, pyruvate kinase, and hexokinase, glucose 6-phosphate of the magnesium, calcium, so copper, potassium, magnesium, manganese, nickel, zinc. Along with that, some molybdenum is also acts as a cofactor in the enzyme of the di dinitrogenase. So dinitrogenase. So these are the cofactors and their enzyme. Now we have to know some of the dietary precursors. Now we have to know about the some of the dietary precursors. Let's talk about the some dietary precursors like. Okay, so like our dietary precursors are like biotin or pentothenic acid, pentothenic acid, and riboflavin, nicotinic, and along with their pyridoxine, folate. So these are the dietary precursors, or they are essential also in our body. So what are the coenzymes? These are kind of important factor for the dietary precursors. So we have to know about the different type of coenzymes like biocytidine, that can help on the biotin, uh, and their chemical group transfer is a carbon dioxide, CO2. So these are the important aspects. So my in that class my main target focus on the enzyme kinetics part. So let's talk about some enzyme kinetics, and then I'll go through all of these. And in before going to that, so we have to know about some of the basic things like the how enzyme works. So enzyme, firstly, for activity of the enzyme, the substrate is very important. So let's write down the substrate. This is a substrate. So substrate, so enzyme binds with the substrate. So what they make the one complex that is called the enzyme substrate complex. Enzyme substrate complex, and then this enzyme substrate complex break down, and then that give the enzyme plus P product. So the enzyme catalyzes the reaction to formation of the enzyme substrate complex, and then specific region on the enzyme having like some, like you can say this is the enzyme, and let's say it's a substrate. So it can bind and form the ES complex. What happened to that? So that's the ES complex. So in that ES complex, what it form is ES complex, and after that this can separate it and form that. So in that form the something product, and enzyme is coming, it's another similar form of previous. So let's say it's a P product P. So like this is a product. Now we can talk about the, like this is the region whether their enzyme and substrates are bind in that region. That is we can called as the active site. This is the active site. So this is the active site of the enzyme. The substrate is the molecule that binds with that active site and of an enzyme by various non-covalent interaction. So in active site, which type of interaction? It will be like non-covalent interaction. So like non-covalent interaction, and some of these ionic interactions, like ionic interactions, and some of the van der Waal interactions, some of the van der Waal doubly van der Waals interactions. So these are the interactions act on the active side of the enzyme when it's bind to the substrate. So when this kind of formation form, so like now we can calculate the some of the energy calculation. Like in the ground state, what happened in the during the formation of ES state? So what happened to that? That is called a kind of transition state, and then we get the product. So it's kind of 2-3 stages are there. Let's draw it. So this is our you can call the reaction coordinate. What happened? Reaction coordinate, and in that state you can call the G. It means the free energy. Let's write down one state. You can say it is a ground state or a state. So in the substrate, when substrate is there, nothing is in the reaction form. In that time you can say that this is in the ground state stage, or you can say that the is substrate form and it's a ground state. So when it is in ground state, when the enzyme and substrate bind, what happens? So in that time the free energy is increasing. So in that time, that is when ES complex form, that time they are in the transition state. In this peak form, it is called the transition state. Transition state. So this is the transition state. In the transition state, whether they are ES to EP is formed. So let's call ES to its form the EP, enzyme substrate state. So it's there, and then in after the enzyme substrate, when you get the P, that is the called product stage. So that is also in the ground stage. That is also ground stage. So in that the transition state, when it is formed the ES to P, there is some similar type of curve like formation of ES, then formation of EP. So kind of two peak are there, ES and EP. So in that time, this is the small changes of this energy, like when this is a ground state and this is also ground state, but in the substrate and the product, so that in between there is some energy gap. You can say that is the delta G0 degree, or there is some energy difference in between that, the when enzyme substrate complex to the substrate or EP substrate, that is the called the del G cat. And the energy changes during this time, this is the complete changes of this. So you can say it the del G uncut. So this is the kind of their reaction coordinate diagram, along with the comparing their uncatalyzed reaction. So like when what will be the uncatalyzed reaction? So in that state there is a kind of products are there still. So this kind of the uncatalyzed reactions, and some of these, sorry so these are the called substrate reactions, and some of these like uh, in the transition state there the energy changes. Now let's talk about the, so in that diagram we understand about the A state and some of A state like surface substrate state and their product state and their energy changes during the reaction coordinate and their free energy. So now we can go through the enzyme kinetics first. So that means let's talk a assuming condition. What will be the assuming condition? So like R, let's take the R is the reactant. R is the reactant, and we call the P is the product. So let's say R is the reactant and P is product. So that means their molar concentration. So like in that condition, reactant to product. So our target is the reactant to product. So that means the molar concentration of the reactant and the product is respectively like, so rate of the reaction can be expressed in the two terms. Let's talk it. So like R equals to R, you can say the reaction rate. So like the, what is the mean of it? The decrease in concentration of R. So during the time first the R is, if the substrate reactant is R, so that means the decrease of the R continuously decrease, because if it is decrease the product will increase. That is a reaction. So that's why you can say it's a decrease. We take a sign, conventional sign of minus. If we will take it del R by del T. So why it is the, because disappearance, you can write down here disappearance of R. So now we are talking about the because it's a decreasing, R is decreasing, so like minus sign due to decreasing and disappearance of the R. Like that, there if something appear is there, R is decreasing, disappearance. Similarly, product is appearing. So you can say that if it is P equals to, product is continuously increasing, the sign will be plus. So like del P by del T, that is the appearance. So I already we have know the main scenario, like this is the, sorry, so this is the things, enzyme plus substrate, that will form their enzyme substrate complex, and enzyme to enzyme plus P product. So that means something is decreasing in that section and something is increasing in that section. Let's talk about the already we have talked about the disappearance of the R, and then the appearance of P. So what happened? So what will be the rate? So technically, the both the rate will be like technically. So reaction rate, if we talk about the, if they are equal, their unit rate of the reaction, like concentration over time, if the concentration is taken like mole per liter, it's called molar, and they like time in seconds, therefore the unit reaction will be like, so what will be the unit of the reaction? Mole per liter per time is, that is the unit reaction. Let's, so what will be there? So that means the rate will be equals to minus del R by del T equals to del P by del T. Now already we got that equation. Now we talk about the, that let's assuming some of the reactions or the rate expressions or rate law that represent that the rate of the expression, rate of the reaction in terms of their reactants concentration. So rate law is known as the rate expression or rate equation. Let's consider a simple equation. Let's write down here. You take A capital A plus B small b capital B. After reaction it can form C capital C. Okay, let's write down in the next page. Let's write down the next page. Yeah, so let's small a small a capital A okay, let's remove it, write down clearly. Small a capital A plus small b capital B. After reaction what will form? C capital C plus D capital D. So let's, this is a reaction, whether ABC is the stoichiometric coefficient of the reactant, and then the rate law, if it is the reaction, then what will be the rate law? The rate law will be proportional to the a to the power x into b to the power y. So whether x y is determined experimentally or may not equal or a to b respectively. So by substituting this value in the rate, like we already get that the minus del r by del t, already we get that in the above reaction. Let's rate, putting that rate in that equation. So minus del r by del t equals equivalent to a to the power x b to the power y. So by removing their, by because it's a removing their alpha, so like alpha is the proportionally constant, whether the k is known as the rate constant. So you can say it, okay fine, then you can write down by replacing, in place of replacing alpha we use the k and we give that the rate what happened to that. Sorry. So like rate constant you can write down, let's say rate constant, k into 1 you can write down it, so it's a rate constant. Then what will be there? So rate equals to k into a to the power x into b to the power y. So what will be the k? K will be the rate divided by a to the power x and b to the power y. So here a b c d you already have defined it, and also I have mentioned it there what is the x and y. So hope that in that portion you will get to know about that. So there is a condition x is, it might be equals to y or y might be equals to x, or respectively it can be there. So let's in terms of units, so it's a rate, k means it's a rate, sorry, k means is the unit rate constant. So what will be the unit of the rate constant? Unit of the rate constant will be like mole l is, in previous we have written that, and now mole by l to the power n. So that might be the different reaction order and different. So let's talk about the zeroth order reaction order, first order reaction and the second order reaction. Let's write down it. If it is the zero order reaction, what will be the type of curve? Like this is the rate and that will be the concentration. So that's a zero order. If it is the first order reaction, then it will be like straight, this is the rate and concentration. And if it is the second order reaction, then this curve will be like that. So it is also rate second order and that is also concentration. So there is the rate constant in the graphical representation of the zeroth order or zero order, second order and the first order reaction. So already we have given that if it is the zero reaction, zero order reaction, then what will be the unit? Unit will be like, already we have written down that in the previous page, mole per L per S divided by mole per L. If it is a zero order, then it will be like 0 in terms of N. So what will be that ultimate? Mole L inverse S inverse. So that will be the units of K. If it is the first order reaction, then mole by L by S by mole by L. So in place of 0 it will be 1. If it will be 1, the mole will be vanish, L will be vanish, it will be like S to the power minus 1. If it is the second order reaction, what will be that? The mole L is divided by mole divided by L, it will be square. So what will be there? Mole inverse L inverse, sorry, L square up to the L and S already by S, so S minus 1. So these are the unit of the K in during the different order of the reaction. So zeroth order, first order and the second order. So what is the meaning of the zero order? So you can see in that curve, their zero order reaction means the rate of the reaction is independent of the substrate concentration. So I can write down in another color. Yeah. So it means they are independent of substrate concentration. These are independent of the substrate concentration. If that is the first order reaction, that means they are one reactant, like rate equals to K. There is a rate equals to K. K means substrate in there's a only time. So that means directly proportional to rate and constant, directly proportional to one reaction. So directly proportional to the one reaction. If it is the second order reaction, that is the either proportional of the square of the concentration of one reactant or proportional to the product of the concentration of each of the two reactants. So that's why it's a second order. Hope that you can understand. Let's going to the enzyme substrate reactions. Let's talk about the kinetics. Yeah. So you can say firstly your this enzyme reaction is Michaelis-Menten equation. So two scientists, one is the Michaelis and the Mott Menten in 1913. So they propose a suitable model for the study of the enzyme kinetics. So they like Michaelis-Menten postulate that the enzyme and the substrate is initially the form the complex of the enzyme substrate complex, and then the breakdown of it, the enzyme substrate complex, to the product. So let's write down it. So like there's an enzyme and then is a substrate, and substrate is like decreasing continuously. That's a rate constant. Let's say it the K1 is rate constant, then it form, what will form, their enzyme substrate complex ES, and it will be some rate constant, you can say there is a K2 rate constant, then it can form the enzyme plus product. So these are the K1 K2 are the rate constant. Technically it will be like someone will be like k minus 1 because that can be decrease decrease sorry rate constant means the velocity will be decreased. So let's talk about the now what we have to remind in our exam. That's very important, that very crucial. So we have to memorize those Michaelis Menten equation. What is it? V0 equals to V max concentration of S divided by Km plus concentration of X. What is the Km? That is Km equals to Michaelis Menten Km Michaelis constant. So what is the Km? Km equals to K minus 1 plus K2 divided by K1. It means the decreasing rate, you can say here K minus, no, not that way, you can write down here K minus 1. So like K minus 1 plus K2 and that divided by K1. This is called the Km calculation. So what is the V0 and what is the V max? And the S means the concentration of S. So in catalytic reaction, if it is a Kcat, then it will be like V max divided by Et. What is the Et? The enzyme total enzyme concentration. This is the called Kcat calculation equation, that is the Michaelis Menten equation, and that is the Km value calculation. Let's talk about the substrate concentration and the initial velocity if in the half of Vmax. So what will be the type of curve? This is the curve, whether this is the plot, like that is the called the initial velocity V0, and this is the like substrate concentration, substrate concentration. So in the substrate concentration it will be like millimolar, and in the velocity, initial velocity will be like micro mu m divided by mu, and in the km, what will be the in km position, the value will be like half of the V max, and that is the of the V max. So this is the effect of substrate concentration to the initial velocity of the enzyme. Catalyzed reaction is shown in that curve. Now another question is come, what is the specificity constant? So for specificity constant, it will be like k cat by k. So like m minus 1 is minus 1, that will be like the specificity constant, and their catalyte, and it denote the catalytic efficiency, like catalytic efficiency. So hope that you will just go through that equation, because you can say some questions may arise in that some of these exam or like, and along with some 2 3 equation like line over bar plot, you just remind that whatever in the line over bar plot. So like 1 by v0 equals to km divided by v max into concentration of S plus 1 by Vmax. This is the linear bar plot. So 2 3 enzymes you should think about the efficiency like kcat value, and then you can go for the specificity constant kcat by km and you get some value. Let's talk about some of the mathematics like what is the substrate of the enzyme catalytic reactions which has initial velocity of 12.62 mole per liter per minute and the maximum velocity will be there, and what will be the Michaelis Menten constant for of 3.88 mole per liter. So in most of the exam your questions, so you just to remind that Vmax into S by Km plus substrate. So in that condition, what are the V0 is there, in then Vmax is there, like initial velocity is there and then their maximum velocity is there, and you then you check out that they are in same unit or not. If it is a mole per liter per minute, and then fine, this they are in the same limit, and if they are Michaelis Menten constant Km is there, so then what will be the, so you have to calculate the engine substrate reaction and initial velocity and the maximum velocity and the Michaelis Menten constant is there. So then you have to calculate the substrate concentration. What will be the, what is the substrate concentration? So for that type of math, you just put that value on that V0 value you got that, and Vmax value you got that, and also you got that the Km value. Then just calculate the value of the S. Similarly, like some enzyme concentration will be there. If it is the enzyme concentration there is there, that means that is the value of Et. Let's talk about that. So we know that the kcat equals to km, sorry, kk Vmax divided by Et, total enzyme, total enzyme. So then so mm, okay, let's go to the previous slide. So kcat by km, that will be like the specificity constant. So if we want to calculate the specificity constant, we have to calculate the kcat and km first. So like, okay, I have written down kcat by, okay, now write down it, kcat by km. So that is our target. So we have to calculate the kcat by km. Now it's given there the k, we given that the Et is given. Et is means their total enzyme concentration. Total enzyme concentration is given in some value, and also Vmax value is there. So you just put down the Vmax value by total enzyme concentration, and then you get some value, let's say it's x, and then that will be the kcat value. Just put down the value on a kcat, and also you get the km value. Also there the provide some km value in molar, so you just put down that y, and you get the answer of the specificity of that specific constant of that engine. So like, this is the specificity formula, and also you know that the Michaelis Menten equation, and in some cases you can go for the lean over bar plot, some of the Vmax and 1 by V0 related calculation. Now we will talk about some of these like reversible inhibitions, and there are some of these curve like that. So reversible inhibitor binds their non-covalently to the enzyme, and their effect of the inhibition is reversed once the reversible inhibitor is removed from the active site of the enzyme. And there is kind of three type of inhibition. One will be like competitive inhibition, second will be like uncompetitive inhibition, and third is a non-competitive inhibition like competitive, uncompetitive, and non-competitive. So let's talk about the some competitive inhibition. Let's say it's a uh, enzyme, and in that pocket, so technically that type of things can bind with that pocket. So like in the active site, other product, like let's say those are in the competitive inhibitor, so they can bind with the active side of the enzyme, they are called the competitive inhibitor. So like in that region, this is the active region, in that active region those are bind with that particular enzyme, and then the activity of this enzyme get diminished, these are called the competitive inhibitions here. So in competitive inhibition, like then the substrate concentration, what will be that? So we know that V0 equals to V max concentration of S, like and Km plus concentration of S. So that we know. Then we also know that they are alpha Km, whether they are apparent of the Km, and during the competitive inhibition their value of the Vmax, the maximum value, the value of that remains the same as the Km with the increasing factor of the alpha. So let's it will be like alpha Km. Whether they are in competitive inhibition, like the in competitive inhibitor, no turnover number of the enzyme. So that means substrate is much more greater than I inhibitor. So what will happen? It gave some formula, and then ultimately we have to just remind the altimeter. So ultimately it will form, like if we put on that 1 by V0, Km in place of Km, it will be like alpha Km divided by Vmax into concentration of S plus 1 by Vmax. So these are kind of Michaelis-Menten equation for the competitive inhibition. So one carp will you can remind of that. Otherwise, you can, okay. Let's talk about the uncompetitive inhibitor. If it is an uncompetitive inhibitor, that means uh, like do not bind the active site of the enzyme. So these are kind of not complete the as a substrate. So that means that can bind with the another side of the enzyme. Let's talk about this enzyme, and you can say, okay, you can say these are the active site, but the this kind of inhibitor bind in that position. So what will happen if it is bind and then its structure gets changed, like structure is kind of changing, that time this binding sequence also get changed, and then the substrate cannot bind to that active site with that same type of affinity. That's a kind of uncompetitive inhibition. So in that inhibition, their presence of the inhibitor diminish the binding of the substrate to active site, but here inhibitor does not bind with the active site. It can bind with the another side of this particular enzyme, and then the structure changes. If the protein structure changes, then this binding site affinity is decreased. So what will be the reaction? Like V0 equal to Vmax by alpha dash into concentration of S divided by Km divided by alpha dash plus concentration of S. What will be the alpha dash? Alpha dash equals to 1 plus inhibitor concentration divided by reaction rate Ki. So what will be the Ki dot, like Ky dot equals to same type of things like Es complex multiplied with I inhibitor complex divided by inhibitor complex and this. So for uncompetitive inhibition, Lineweaver plot will be like that type of curve will be there, and in that section 1 by minus k. So these are the uncompetitive inhibition. So some of these like mixed inhibition, like in the active side some inhibitor also bind, also that bind with the another side of the specific enzyme, that kind of things like the mixed inhibition of that specific enzyme. If in special case for non-competitive inhibitions like uh, if Vmax is decreases but Km each remains constant, so Vmax diminish but Km is remain constant, that is a kind of pure non-competitive inhibitions. So reaction the formula will change little bit. Let's talk about the uh, 2 3 plots are there. If I think it's fine. Now we can talk about some of these reversals in irreversible inhibition and some of these curve nature in that sense. So you can know, you know about some of this like irreversible inhibition, like some substrates in some enzyme kind of tightly bind with their activity, and if some of this protein get bind with that kind of enzyme and they are like kind of fixed, so like that's a kind of inversal, like irreversible, sorry, so kind of irreversible. And some of these inhibitors or inactivator kind of suicide inactivator, these are type of irreversible inhibitor that can comparatively unreactive, but until they would bind to the active side of the enzyme, due to the structural complementarity they are bind to the active side and then get converted into highly reactive compound, and that results the death of the enzyme. That's why it's, you can say, called the suicide inactivators. So some of the suicide inhibitors are kind of aspirin and the penicillin, and also allopurinol. So these are kind of the suicide inhibitors. So let's talk about this with the structure, how aspirin bind. Let's talk about the structure. Now I can write down the structure of the aspirin, like COOH bind with the oxygen, C double bond O, CH3. So this is the aspirin, and that bind with the one of the key enzyme that is called the cyclooxygenase 1 or COX1. What will be the structure. In that structure you can set some of the serine molecule is there, and then CH2, then it will be like OH. So when it is bind with that uh, molecule like aspirin, then is called active, then it form the salicylic acid. How what will be the structure? COOH, and that will be like OH. So salicylic acid, and if it is form the salicylic acid, that can inactive the cyclooxygenase enzyme coxon. So these are kind of aspirin. And now talk about the penicillin. So for that penicillin, it's an antibiotic which inhibits the enzyme glycopeptide transpeptidase. Glycopeptide transpeptidase. So let's transpeptidase has such structure like trans. Sometimes what happened? Let's take another color. Transpeptidase. This is called transpeptidase. And then it has some structure of protein called serine, bind with the O and bind with the H. So then H will remove in that section, then it will bind to the penicillin. So let's write down some of this ring. Nitrogen is there, one will be CH3 group, CH3, and in section it will be like COOH, and in that region it will bond with C oxygen, and then NH, C double bond O, R, and in that portion also it binds to the C double bond O. So exactly in that position, sorry, it's a C double bond O, yes. So in that double bond it will attach, and then that will also form, and then this bond to that side. So that form that the transpeptide is serine to that region, and this is the penicillin that bind with the transpeptidase enzyme or glycopeptide transpeptidase, and then it's form the transpeptidase. Like this portion, this serine portion get attached to that region and form the transpeptidase. So you can understand how the inhibitor bind with that enzyme and how they inactive the activity of the enzyme. So there is a kind of basic uh, class. Hope that you guys can understand about that. What are the incident going behind this, and if you want to know about more detail, I can also give them how these kind of equations are coming, because that is not so important for that exam purpose and all, but you can evaluate all those things. Then I can one class I can explain all how these equations are coming, or the derivation of the equation and how they formed. One class I can explain it, and so in that class this is my kind of brief things, and I want to cover all those aspects, and how we can remember all those things. That's why I'm just taking this class. So enzyme kinetics and how it is coming on, and what are the rate constant and how it is derived on, and then the major things are Michaelis Menten equation and their kcat or kcat Vmax Et total enzyme concentration, and then their specificity of this specific constant kcat by km, and how we can calculate of that. And also I have calculated the dimensions, like how rate constants, what will be the rate constant, and if it is a first order reaction, if it is a second order reaction, or it is the zero order reaction, how it will be there, and who, like in the zero order they are independent of the substrate concentration, and first order and second order they have their another aspects. So hope that I have discussed it very briefly, but later I can also go through the incomplete details, and also I can complete details of these, some of these, like different say cycles, the role of the metabolism, like of the TCA cycle and their uh, urea cycle, and along with some the how the energy is produced in our cell, that is also important part, and how it is going on in one class. Hope that I can explain about that, and some after that I can explain some of these, like malate aspartate shuttle and how it is works on that, and then some of this like glycerol 3 phosphate shuttle. Okay, hope that you guys can understand of that class today. Bye bye, take care, good night. I just turn off that stream.